That's good. Okay, let's get started then. Good morning. Good, you're awake. Okay, so let me tell you a few more stories about structural biology and drug discovery, or structural biology helping us understanding how small molecules work to regulate proteins. Okay. So another big story is big family is protein kinases. Are you familiar with protein kinases? Somewhat. Okay, well, anyway, either way. So I'm teaching, so I will have some background information here, so please bear with me. So protein kinases, as the name suggests, phosphorylate the protein. And the phosphoreceptor is either serine, threonine, tyrosine, or rarely histidine can also be phosphorylated. So ATP is a donor of the phosphate, so they all contain ATP binding site because they all need to bind ATP to do the catalysis. And the kinases are classified in two groups: serine threonine kinase and tyrosine kinase. Because you can sort of imagine the side chains are quite different. A serine side chain and threonine side chain much smaller than a tyrosine side chain. So the kinase, even though they are homologs, they are a little bit different from each other. But there is a subclass of protein kinases that can phosphorylate both serine and tyrosine, the so-called dual specificity protein kinases. But most of them do only serine or tyrosine. So it's a large family in humans, about 500 protein kinases, roughly 1.5% of the human proteome or genome, the genes. And they all have very important cellular and biochemical functions, controlling many processes, phosphorylating their substrate. And then when dysregulation kinases are associated with many diseases, especially cancer, you see. And then, so they are very important targets for drug discovery. They're also very successful targets for drug discovery, as I'll show you. So the 500 or so protein kinases that are sequence-wise, they're homologous. So you can build a tree for them based on sequence homology. So that's the tree right here, the human kinome. So the 500 kinases here, they are broken down into different kinds of kinases, this and that. So I guess that's all I'm going to say about that. And kinases are important for drug discovery because you can see, as of 2021, now 4 years ago now, 60 compounds have been approved for human therapy. They target various different protein kinases. The disease area primarily cancer and then inflammation. So today you'll hear a story about inflammation and a story about cancer. Okay, so as you can see, most of these compounds has the nib at the end. If you look at the names of the compounds, and that's sort of the indicator that these are all protein kinase inhibitors. They all have a nib at the very end. A small molecule inhibitor or protein kinase is something nib. Okay, so now that's the intro. So now let me tell you about the story of my life. Well, my life in a pharmaceutical company. Okay. One of the projects I worked on started with this paper, which came out at the end of 1994. So nowadays, protein kinase inhibitors are well understood. They're well accepted. This is 30 years ago. There were a lot of questions about how you can get an inhibitor against a protein kinase. I'll explain why that's the case. But anyway, so this paper came from SmithKline Beecham Pharmaceuticals. You probably never heard of them because you're too young. But you've heard of GlaxoSmithKline, right? GSK. So essentially, the Glaxo bought SmithKline, and they merged together, and now it's called GSK. Okay. Anyway, so what they found essentially is a protein kinase that's involved in inflammation. So that makes this compound, this target, for anti-inflammatory discovery. So because of this paper, many companies in the U.S. or probably around the world are working on anti-inflammatories. Remember JTE 607, 1999, anti-inflammatory? It's probably part of the same wave. So how did that discovery happen? So it was known for a long time that if you challenge human monocytes, immune cells, or THP1 cell line with LPS, you know about LPS, lipopolysaccharides, bacterial cell wall component. If you challenge them, the cells with LPS, the cells will secrete TNF and interleukin-1, tumor necrosis factor and interleukin-1, pro-inflammatory cytokines. And then scientists at SmithKline realized actually if you pre-treat the cells with this compound, as for example, many compounds like in this class called pyridynyl imidazoles. Okay, pyridynyl imidazole. So that's the scaffold that defines this class of compound. And this is the most famous one, sp203-580. You'll see this a few times a day. So if you treat the cells with this compound first, and then challenge with LPS, they no longer make TNF and interleukin-1. So basically, you downregulate the production of pro-inflammatory cytokine, you downregulate inflammation. So you could sort of think about this compound could be good for anti-inflammatory uses. Okay. So all of these are cell-based assays. The compound works against the cell, but how does it work? It's not known. So what's a TNF? Are you familiar with TNF interleukin-1? You are? Then I'll skip over this slide. Okay, good. Yes? Good? All right, moving on. So again, this is 1994, published 1994. The work was done 1992, 1993. So 30-some years ago. So they used a compound that contains the radio-labeled iodine 125. So put a radio label and put a azido group on the compound so that it's a photo label as well. So then they they incubate this compound with a mouse lysate, cell lysate, and then shine UV light on it, and then run a 2D gel. You don't know what 2D gel is? Oh, it's okay. Anyway, this is classical ancient way of cloning a gene. Okay, you probably will laugh at it now. Anyway, so when they do this, they find only one spot that carry radioactivity. That suggests the compound is quite selective. I mean, you're basically incubating against the entire lysate, and then you turn this on to turn that into a free radical. You should be able to bind to anything. If whatever target binds to, you should label it. But only one spot carries it, so that means there's really one important target in the cell. That shows the selectivity, which we'll come back to. And then once you have that spot, back then, there's no other way. You cut it out. You cut the spot out of the gel. You do peptide sequencing and then you clone it from using primers, you clone it from a library. Nowadays, of course, you do it completely differently. You do mass spec, and you find the protein right away. But anyway, this is how they cloned it. Before I show you what before I show you what that is, let me show you briefly signal transduction, which are you familiar with this? If you are, again, I'll skip over. Well, anyway, so you have extracellular signal binding to a receptor, which is a tyrosine kinase. The tyrosine kinase phosphorylated tyrosine, which recruits GRAB2, SOS, activating RAS. We'll talk more about RAS later today. So GTP is bound to RAS, then that can activate RAF, which is a kinase, and it goes to something called the MAP kinase cascade. Okay? And that's shown right here. So in vertebrates, you have what I just showed you, RAS, RAF, MKK 1 and 2, and ERK. So this is for cell growth. Okay, it's a cascade, so you have MAP kinase. MAP, it means mitogen-activated protein kinase. Okay, you have a kinase, and above that, you need a kinase, so phosphorylated kinase, there's MAPKK, and then MAP3K, MAP4K, MAPKKKK. Okay, so you have for growth, for stress, hyperosmolarity. The protein they found is P38. So P38 is a target for pyridyl indazole compound. And then these pathways are also conserved in Cerevisiae. So P38, MAP kinase. So it turns out actually there are 5, P38 proteins in humans. So P38 has 2 isoforms, which are splicing variants. Obviously, that's the one that's sensitive. That's the one they found that's sensitive to the compound. And then later was found there's also P38 beta, 74% identical, also sensitive to the compound. And then you have P38 gamma, 63% identical, but no longer sensitive. So even though it's highly homologous, but it doesn't care about the compound anymore. And then finally, P38 delta, 61% identical to P38, also insensitive. So in fact, actually, people have screened this compound, SP203-580, against a panel of protein kinases and also a protein phosphatase. So you can see the compound is active against P38, IC50, 50 nanomolar or so. And then against P38 beta, IC50 is about 100 nanomolar. So both alpha, or you can call this alpha, that beta, is sensitive. Gamma, even at 10 micromolar concentration, the enzyme still inhibits only about 25%. So much less sensitive to the compound. Okay? And then ERK and junk. Oh, forgot junk. Well, it's okay. So these are the other 2 MAP kinases, ERK for cell growth, junk for stress. Even at 100 micromolar concentration, you basically have barely any inhibition. And the same is true for other protein kinases and also the protein phosphate. So the compound actually is highly selective against P38. So how familiar are you with protein kinase structures? Okay, no answer, that means no, I guess. Okay, let me go through that a little bit for you. So here is protein kinase sequence. Here is protein kinase structure. Typically, a protein kinase domain contains about 300 residues. And at the sequence level, there are many motifs, so 11 motifs here. And each of these motifs have important roles in binding, catalysis, and everything else. So you have this, especially you'll hear a lot about this DFG motif, which, as you see, chelates magnesium, that's important for catalysis. But you see that in a different context. So all of different motifs at the sequence level, and then at the structural level, you have secondary structures, and then also you can see the structures, 300 residues, actually is 2 lobes, 2 domains, or in the kinase jargon, lobes. A large lobe and a small lobe. And the lobes can go this, which you saw that yesterday already. So in the small lobe, it's mostly beta, 5-stranded beta sheet, and 2 helices that are named alpha B and alpha C. And alpha B could care less about. Alpha C you'll hear again and again today. That's a very important helix for kinase activity. And then large lobe, a few small beta strands, are mostly helical. Okay? And then obviously there's a linker between the lobes right here. And this linker also you'll see today, very important. So here is a schematic structure of a protein kinase, large lobe at the bottom, small lobe at the top. That's usually when you show a protein kinase structure, this is the canonical view of it. Large lobe at the bottom, small lobe, the beta sheet, alpha C helix right here, going into the screen. So the conserved residues are shown right here. And then ATP, you can see, is bound between the two lobes. So the adenine bound deepest, phosphate on the outside. Okay, I'll come back to this a little bit more later. And then the adenine, you can see, contacts the linker between the two lobes. So the adenine actually is recognized by the linker between the two lobes, the main chain of the linker between the two lobes. And then I can say a lot more about protein kinases, but anyway, for most protein kinases, as you saw earlier, require phosphorylation to be activated. And then phosphorylation site sits in something called activation segment, that's shown in magenta here. Start from here down to here. And that segment is bracketed by the DFG APE conserved motifs that I showed you on the earlier slide. So these two motifs bracket a loop that's quite variable among protein kinases, containing different kinds of phosphorylation sites. So that controls, once this is phosphorylated, then the kinase can be active. If it's not phosphorylated, the kinase is not. So that's example that's shown right here. So these are various different protein kinases, ERKs, JUNKs, P38, ESHOC1 is the homolog, CDK2, CAPK, SAC-AMP dependent protein kinase, which I'll show that next. Anyway, so DFG marks the N-terminal end of this motif of this activation segment or loop, and the APE, alanine, proline, glutamate, is a C-terminal end. And then shown in red are the residues that need to be phosphorylated. So MAP kinases need to be phosphorylated on the threonine and the tyrosine in this loop before they can become active. And then CAPK require a threonine, a single residue, need to be phosphorylated. Okay, so we will mention this briefly, but DFG is something you'll hear a lot more about. Okay, so the first structure of a protein kinase was the structure of CAPK, SAC-AMP dependent protein kinase. So as the name suggests, it depends on SAC-AMP. So this is the protein kinase subunit of the entire holoenzyme, which contains 4 subunits. But anyway, so you have ATP bound, as I said earlier, between the two lobes, adenine bound deepest, phosphate on the outside, and also there are 2 manganese ions in green, that chromin is the metal, the phosphate on the ATP. And this structure also is in complex with a peptide inhibitor, which you can see start from here, helical structure, and then a long loop that goes across the active site. Okay? If you look at it carefully, there's this alanine right here. Do you see that, the alanine? So if this were a true substrate, this will be a serine. So the serine can then attack the gamma phosphate and become phosphorylated. So this structure tells you exactly how the kinase works. It binds ATP, but adenine deepest, phosphate on the surface, so the phosphate can sense or can see the substrate amino acid residue, serine, tyrosine, threonine, and then that side chain is activated by a general base to extract the proton, and then the oxygen attack the phosphorus. That's what you get the kinase reaction. Does this all make sense? Okay, so, and then this is how this helix is called alpha C helix in protein kinases, because the first structure named all the secondary structures, and that basically gets stuck. People just use the same nomenclature going forward. So that structure is in a closed state because it's bound to ATP. So the small lobe closed down onto the larger lobe, so the ATP is bound in the middle. And then ERK, the MAP kinase involved in cell growth, That structure, one is on its own, is in the open conformation. You can sort of see the small lobe now peels back. You can compare the two. You can see the difference. Small lobe peels back. So there's a big gap between the two. So this is the inactive state. Okay. The other thing you can see by comparing the SUC-AMP-dependent protein kinase and the ERK is that ERK has an insertion, extra paired little helices at the lower right, which does not exist over here. So actually, this insertion occurs in almost all MAP kinases. Why it's there, we don't really know. But that's something unique. So even though these are homologous, there's some unique features among these enzymes. Okay, so now to look at how ATP is bound. So adenine is recognized directly by hydrogen bonding to the main chain amide to the N1 atom and the main chain carbonyl to the N6 atom. So you all know about AU-AT base pairs. That look exactly the same, right? So this way the protein specifically recognizes an adenine base. And then N7 has a hydrogen bond with a threonine side chain. The ribose hydroxyls has some hydrogen bonds, and the phosphates are involved in a large network of ionic or polar interactions. But this, as you saw earlier, is on the surface of the protein. So as you see later, it doesn't really add too much to the binding affinity. So the main thing you want to remember is essentially the adenine is recognized by hydrogen bonding for specificity, and also two lobe closes down, gives you a lot of hydrophobic interactions of the base itself. So if you look at the binding affinity, again published many years ago now, of various ATP homologs for AMP, SUC-AMP-dependent protein kinase. So ATP KD is about 10 micromolar. So pretty good affinity. A-bromo is fine. ITP, 14,000. So, and then GTP, 3,000 or 4,000. So you can sort of see selectivity. The enzyme recognizes ATP, it doesn't like GTP, even though it's also a purine base. And then DATP, you remove the 2' hydroxyl, doesn't matter. Remove the 3' hydroxyl, doesn't matter. And then SUC-AMP, not too bad, but not as active. And then now you start chopping with the phosphate. AMP CPP is basically with a carbon as a linker between alpha and beta phosphate. That's the same as, almost the same, it's only threefold difference with ATP. And then you start chopping. ADPCP, this is another ATP. ADPNP, another ATP. ADP itself, same as ATP. So you don't really need a gamma phosphate binding affinity. AMP is a little bit worse for some reason, we don't know. And then adenosine, no phosphate at all. It's threefold weaker than ATP. So essentially, the phosphates, even though it has huge number of interactions, doesn't confer binding affinity that much. The main binding affinity comes from ATP, which is somewhere around, from the adenosine part, which is about 30 micromolar or so. Okay, so I was working at the company, Boehringer Ingelheim in Connecticut, and we saw the paper. Well, I didn't, the company saw the paper, and then everybody jumped in to this target P38 for anti-inflammatory drug discovery. So I'm a structural biologist. The idea is to determine the structure of this compound bound to the enzyme to understand how it works at a molecular level, and use that to drive drug discovery. So here's the SP203-580 compound. I showed you many times. And the chemist, to help me, made two additional compounds, putting an iodine this position, which you saw earlier, that's where the radio label is, right? So they put the iodine here, or put the iodine at the 4' position. This is the 3' position, this is the 4' position. The reason they did that because, as I mentioned earlier, when you talk about crystallography, the heavier the element, the more electrons there are, the easier, the stronger the diffraction. So this could be used as a marker to help us locate the compound. It turns out having this also helps us getting better crystals. Well, you take what you get. So we have a structure of this compound bound to P38. We have crystals, I guess, beautiful looking pictures. And so here's the structure. Large lobe at the bottom, small lobe at the top. The compound binding right here between the two lobes. So that's where ATP binds. So right away it tells you that the compound competes against ATP. That's how it inhibits, because the ATP cannot bind anymore. But to our surprise, you see, I showed you that little insertion in the MAP kinases. A compound is bound here as well. We did not expect that at all. So only when we saw the structure, realized actually there's a compound here as well. It binds very nicely right here. So that's the insert is right here, the insertion is right here. This is the body, the large lobe, and then the compound stuck right here. The iodine sticks deepest into the pocket. So that's a real pocket here. A compound can bind, but we have no idea what the function of that binding site is. And basically, we did not keep on looking, because at the company, you look at only the part that drives drug discovery. This one we don't understand. So hopefully one of these days we'll figure out what that insertion does and what this compound binding here will do to the enzyme. But if you look at the ATP binding site, so you have large lobe, small lobe, the compound inserted deep into the interface between the two. Here the linker segment between the two. So then if you look at the overlay of the binding mode between the compound, which is showing cyan, and binding mode of ATP, that's expected. Okay? So you can see, sort of explain how everything works. For example, the pyridyl group here, the pyridyl nitrogen sits essentially the same place as the N1 of adenine. So this, you can imagine, then maintain the same hydrogen bond to the kinase. The hydrogen bond from N6 of adenine is sacrificed. There is no hydrogen bond for it. At least for this compound, there is no. And then the imidazole sort of, pyridyl imidazole together sort of mimics adenine base. And the rest, this ring here sits where the phosphate is. Okay? So looking at this, you can explain something. When you do drug discovery, you hear that a lot, structure-activity relationship, SAR. You've probably heard of that. So basically, you change the compound a little bit and see what happens to the activity. So in this case, this is, again, scientists at the SmithKline. So the pyridyl group, they change the nitrogen from the para position to the meta position to the ortho position, just basically marching the nitrogen around the ring. You can see at the para position, very potent. And once you move the nitrogen around, you lose more than 500-fold affinity, because it shows the importance of the hydrogen bond to the protein. So that's very nice to have. And then the other thing you see is this group here sits where the phosphate is. I just showed you the phosphate doesn't give you binding affinity. So, and that's also shown by SAR from the company. So in this case, they use another series compound where the substituents come from the N1 position rather than the C2. So essentially, you can see they put in all kinds of different things here. The activity doesn't change much. You can have only a hydrogen there, nothing there. You have only a twofold or so loss in activity. So that's basically shows you don't really need anything here, but they are here because binding affinity is only one aspect of developing a drug. There are many other things you need to do. I'll show you that again today, later. So these substituents maybe improve other properties. May not improve the binding affinity, but may improve other properties of the compound. So I've been talking about the compound here against P38, but you may remember I said at the beginning there are 500 protein kinases in humans, and they all use ATP as the donor of the phosphate. So you may start wondering, and that's what people were wondering back then, how do you achieve selectivity? That means there are 500 ATP binding sites in these 500 kinases. How can your compound go in and hit only one of them if you compete against ATP? You understand the question? So that's the question back then that people think it's going to be very hard to develop protein kinase inhibitors that are selective. If you develop a compound that kills all protein kinases, well, it's probably going to be toxic. You don't want that. So the question is, how do inhibitors achieve this selectivity or specificity against the individual kinase? And once you understand that, maybe the better thing to do is how can you develop such compounds against a kinase that you're interested in. So now we have a structure of the compound bound to P38. So the next question we want to answer is, how does this compound achieve selectivity? As I showed you earlier, the compound is quite selective against P38. So now if you look at the binding mode compound, so this part mimics ATP, right? The pyridinol imidazole plus this phenyl ring and the sulfone mimics ATP. But this 4-phenyl ring, you can see, sticks out from the ATP envelope. So the hypothesis will be then this 4-phenyl ring senses a different part of the protein kinase that ATP doesn't care about. And somehow, there is something special about P38 that allows this large phenyl ring plus the iodine to sit there. That's the idea. That sort of makes sense. So now with that idea, we went in and looked at it in detail how this ring is recognized, how it's bound to the protein. So here I overlay two structures, P38 in green and ERK2 in magenta or purple. What did you say? Pink. Dark pink. Okay. All right. Pink. Okay. So this is the alpha C helix. Remember alpha C helix that I talked about many times? So alpha C helix contains a glutamate, which unpaired with the lysine that come from the small lobe. These residues are strictly conserved. Two of the motifs among the many. So that's what they do. This unpaired interaction holds alpha C helix in place. That's what it does catalysis. And then if you look at the ring here, so one side of the ring is the lysine, the conserved lysine. So that's the same among all protein kinases. On the other side of the ring, in ERK, is glutamine. In P38, is the threonine. So you may know your amino acids. Threonine is smaller side chain than glutamine. So by this analysis, we feel that essentially maybe having a threonine here is the trick that allows P38 to be sensitive to a compound with a group here. Okay? So that's the idea. Is that true? Now you go back and look at the sequences of protein kinases. That threonine is right here. This is the linker segment, as I mentioned, between the small lobe and the large lobe. So that threonine is right here. So in P38 threonine, in ERK, CAPK is glutamine, as I showed you earlier, or methionine. Actually, methionine is one of the most popular amino acids at that position. So these two enzymes are sensitive. Remember, P38 beta is sensitive. You have the threonine. GABA and delta not sensitive. They have methionines. So I think this is a very nice now idea that you have a small residue here that allow a pocket to be formed there. And that pocket can be taken advantage of by a small molecule compound that normal adenine base would not see, would not care. So that's how you achieve selectivity. Unique properties of that kinase. And then threonine does exist in other kinases. For example, RAF1, I'll talk about RAF a little bit later. Threonine, Sark tyrosine kinase also has a threonine. So there are small molecule inhibitors against them that use this threonine as well. So this gives you one mechanism to achieve selectivity among protein kinases. And I also mentioned this linker is very important because right next to this threonine, this is histidine, this is methionine, the main chain recognize N6, so the main chain carbonyl recognize N6, main chain amide recognize N1 by the 100 bonding interaction. So you have basically, this linker has 6 residues, or 7 residues, 6. And then 3 of them have very important roles. Okay. And Kaban-Shokart called this threonine the gatekeeper residue. It's a gate for that pocket. So if you have something big, the gate is closed. If something small, the gate is open. And I'll come back to that a little bit. So the idea further supported when you overlay the binding mode of various protein kinase inhibitors. See, they all bind so that there's a hydrogen bonding acceptor right here, which mimics the N1 of adenine. And most of them actually sacrifice N6 for whatever reason. But you'll see later there are also kinase inhibitors that use both. And then the rest sits where ATP is. So the 3' although the 4-phenyl ring here is the only one that sits over there. So again, it gives you all of these. The other ones are non-specific. They are basically inhibit all many protein kinases. And the sp203,580 is the only one that's selective, and that's the only one sitting out here. So based on this, we have an idea. So this is ATP, adenine ribophosphates, donor acceptor, the N1, N6. So your compound need to have at least one of these to give you binding. But then more importantly, you need something to stick out from the overall envelope of the ATP so that you can achieve specificity, selectivity. More importantly, this binding will give you also potency. Because I showed you earlier, ATP is only about 10 micromolar. That's not good enough. You need to go down to nanomolar. So this group, having this group here also give you additional potency. So that make it into a drug. Okay. So once you make a rule, you know nature is very resourceful. So you can say there might be other modes of specific inhibition of protein kinases using the ATP binding pocket. And I'll show you one right away because we found one right away. Okay, before I show you that, so Kavan Shokhat, I mentioned, now at Berkeley, he was at UCSF. So he came up with a brilliant idea. As I said, so these kinases have methionine in them, so they will not be sensitive. So what we can do is, he can mutate that to a threonine. And then put that into the cell, and then knock out or knock down the endogenous enzyme. So now your kinase now has a threonine, the wild type enzyme. And then he can screen against a large collection of bulky adenine analogs. So those can become ATP or ATP analogs. They can bind to this mutant kinase, regulate its activity, control its cellular function. So using small molecule control almost any kinase you want in the cell by introducing the mutation in this gatekeeper residue. So he called it the bump and hold method. It has been very powerful. Many people actually use it to sort of analyze the function of proteins in cells, protein kinases in cells. Okay. So far okay? All right, so now we have a structure of the Smith-Klein compound bound to p38, but at the company, you don't want to work on other people's compound because you don't have IP on that. You need to have your own compound. So at the company, we did a high throughput screen. I guess you now know what that is now. So thorough, I don't know how many compounds we went through, probably a couple 100,000 compounds against the protein kinase. And they found this as one of the many hits they found. Okay? And this is a diarrhea urea group in the middle, two error groups on the side. And this compound actually is 1 micromolar. For HTS hit, actually is very good quality, very good affinity. So the question is, how does this compound work? So given what I just showed you, we need the hydrogen bond acceptor. Well, that's right there, right? So you could make a docking like this. The hydrogen bond acceptor sitting where the pyridynyl nitrogen is, and the rest sits along where the compound is. Maybe. But of course, you really want to get the actual structure. So we succeeded in determining the structure, and that's shown right here. And to our surprise, again, same view, large lobe, small lobe. So, and then ATP bonds here, as you see, the compound bond next to ATP has no overlap at all with ATP. So then the question is, how does the compound bind like this, and how does it compete against ATP, because it doesn't crash against ATP. So it's a completely unexpected discovery using that compound. But then if you overlay the binding mode of the compound, the compound 1, the high throughput screen hit, against the Smith-Klein compound and ATP, you see essentially the compound has no overlap with ATP, but it does sit in the same region as this phosphorylation, which, as I showed you earlier, confer selectivity. So this compound actually is also selective against p38. But the compound doesn't talk to ATP at all. So how does it work? So I showed you this slide earlier, and the only thing I want to point out right now is this DFG motif, which is highly conserved coordinate magnesium ion. It marks the beginning of the activation loop. So strictly conserved among protein kinases. You have a phenylalanine and a aspartate. Phenylalanine, highly hydrophobic. So how did the compound work? So here, essentially, is a movie. Large lobe at the bottom, small lobe at the top. DFG motif, aspartate, phenylalanine. G is disordered. We don't see it, so we only see D and F. Okay, so this is the carbonyl oxygen of phenylalanine. Glycine will be pointing towards you, but disordered, we don't see it. Okay, so you can see the phenylalanine sits in a very hydrophobic pocket between the two lobes. Okay, all right, and here you have ATP. So let me start the movie. So first, the Smith-Klein compound binds where ATP binds, so it'll compete with ATP away. So ATP is gone. Okay, so that's how it works. Well, my postdoc made this movie some years ago. Anyway, so ATP again. It turns out for the compound 1 to bind, DFG had to rotate. And then that leaves this huge pocket where phenylalanine used to sit, and there's where the compound goes in. So DFG rotates out of the way. And more importantly, it will go around. More importantly, when you rotate out of the way, you'll see actually the phenylalanine side chain clash into the ATP. The compound doesn't care about ATP at all, but the protein now competes against ATP. So that's shown right here. So phenylalanine comes over, sits where ATP is, and then ATP is competed out. That's how it inhibits. So it's an indirect competition by inducing or stabilizing a conformation of the protein that is incompatible with ATP binding. Okay? So again, this is the active site of the enzyme. So another example of a conformational heterogeneity or flexibility near the active site that allows some molecule to bind to regulate the activity of the enzyme. So this is another example. I showed you an example earlier for ACC, the herbicide. So this is another example of that. So now the compound binds right here. Okay? So when we were writing the paper, I need to define this conformation. It's always a mouthful to say it. I say, okay, I need a name for it. So I call it DFG out because DFG is out of the regular pocket. So DFG out conformation, and now actually that term stuck. People in the field actually use this term now, DFG out. And then this normal form conformation would be DFG in. So DFG in, DFG out, the two states of the kinase for the DFG motif. Okay? So if you look at it more carefully, DFG in, DFG out, mostly is the aspartate. You can see assume a different conformation in the main chain. Phi angle changed by 180 degrees. So here is left-handed helix. Here is a beta strand. So the phi angle flipped. That's how you change the state. So this is DFG in right here. Aspartate is right here. The whole thing flips. Then DFG out is over here. And it's clash with the inhibitor, of course, which also the Smith-Klein inhibitor, which also clash against ATP. And then this lysine glutamate ion pair, actually the glutamate now swings around a little bit, recognize the urea oxide nitrogen, forming two nice bidentate hydrogen bonds with the urea group. Okay. So if you look at this compound again, the T-butyl part of the compound sits where phenylalanine used to sit, highly hydrophobic. T-butyl is beautiful for that binding site, so it sits there very nicely. And then the rest is a scaffold. And then the urea nitrogen is hydrogen bonded to the glutamate. And then this part sits where the phosphate ring I showed you earlier, so that gives you specificity. And then we also know that the adenine should be binding over here. This compound does not talk to adenine at all, but there is an adenine binding site over here, so which we're not making any hydrogen bonds to that region. So we thought about, okay, maybe we can change the compound a little bit to grow something here to reach that adenine binding site to give us better activity. So that's done using this compound. by growing something over here into a morpholino, and you see this basically, I'll show you actually 100 bonds to the enzyme. So this compound actually 0.1 nanomolar KD, very, very potent. Compared to the HTS lead, which is 1 micromolar, so it's about 10,000-fold enhancement in affinity. You can sort of see the difference between the compounds, and then based on SAR, putting a phenyl ring here give you about 140-fold enhancement in affinity. And putting a naphtha rather than a phenyl give you about 15-fold. And having this morpholino at the end give you about 11-fold. So each individual modification give you enhancement in potency. And if you multiply all together, it's about 20,000-fold enhancement. But the total is about 10,000-fold. So there's not completely additive. There's a little bit of penalty along the way. But still, it's very close, within a factor of 2. So this is our lead, and this lead hit from the HTS, and this essentially became a clinical candidate that the company took into the clinic for anti-inflammatory. So we determined the structure of this compound bound to P38, shown right here. You can see now this morpholino group reaches over and then 100 bound to the main chain amide, just like the N1 of adenine. So we have achieved that interaction, but then you can see the urea group now recognized differently. The glutamate now, again, now only form 100 bound with the urea because the rest, because this naphthol group here causes a little bit of reorganization of the binding mode here. But overall, binding mode is quite similar between the HTS hit and the drug lead, the clinical lead. And then we have a biochemist or biophysicist who measured the binding on rate and off rate of the compound. So the Smith-Klein compound binds very, very fast, milliseconds, 0.1 second, it achieves full complex already. Very fast on rate. Whereas an analog of this the lead compound I just showed you binds very, very slowly. Takes hours to get on. So I guess this is consistent idea that for this compound to go on, DFG need to rotate out. And P38 doesn't like to have a DFG out. So it takes a long time for the protein to sample that conformation, and then the compound can go in. That's probably what happens. So then if you look at the tables, K on is about 100-fold or more lower. K off also very, very slow. So it goes on slowly, come up slowly. That's why overall you have a KD about, this is comma 1, not 796, K on, and K off is extremely slow. So it goes on very slowly. Once it's on, it's stuck there. If you use this as the K off rate, then you can calculate the T half is about 23 hours, which for a drug candidate is a good thing because you can take a drug molecule and then it stays on there for 24 hours. So you don't have to take drug all the time. Once every day or so might be okay. So that's a good thing to have. So that gives you overall KD of 0.1 nanomolar. So lower K on, much lower K off, give you a very potent compound. So the compound went through phase 1 clinical data at Boehringer Ingelheim. Everything looks good. And then later it went into phase 2, and that's when you encounter toxicity and other things. So at the time, it was not clear whether toxicity is due to this compound or due to the mechanism that is inhibiting P38. So as you can imagine, because of that paper, because what we have done, what we did, there are many, many P38 inhibitors that went into the clinic, and they all failed at the end. So probably there's something you cannot do to P38, but that remains to be seen. So I showed you this slide earlier. You may remember. So I'll just show you again. So we have a very potent compound, very selective compound. Everything else, oral bioavailability looks good as well. But in the end, probably, so there are many hoops you have to jump through to make a drug molecule. And these are the easy part, very easy part. And the bottom part actually is much more difficult. Okay. All right, so that's that story. So, but there are protein kinase inhibitors that use this mechanism. So, so there's an inhibitor of a Abelson tyrosine kinase. It's called Gleevec or imatinib for treating cancer. That's this compound right here. So that's used in the clinic for treating the CML. And then the scientists use the analog, which is this one right here, missing this ring here, to determine the crystal structure of this compound bound to this tyrosine kinase. And if you look at the binding mode, so our structure in green, and then Gleevec in cyan, you can see actually very similar. So they use the same pocket. T-butyl is where DFG pocket is. They use the same pocket. So everything looks very, very similar. Actually, you saw this paper was published in 2000. Okay, so and then our paper came out 2 years later because I could not publish. I saw the DFGL conformation in 1997, but couldn't publish it. But anyway, minor detail. So if you look at Abelson tyrosine kinase, it has a threonine. So it looks like threonine is a magic bullet that allows you to take advantage of that pocket to give you small molecule inhibitors. Okay, so now the other one is RAF, right? So that's the story about P38. So now I'm going to change gear, talk about RAF inhibitors. These are against cancer. So, and this is the V600E mutant found in many melanomas. So it's a melanoma target. So this work was done 20 years ago. So people, well, by my PhD advisor, actually. He formed a company, and that's what they did. So they are much more directed in terms of their screening effort. So they use a collection of smaller compounds, only 300 or so Dalton in size, only 20,000. They use that at a very high concentration, screen against a panel of protein kinases to see which one can inhibit the kinase, can bind to the kinase. And then they determine many, many crystal structures of these active compounds against some various different protein kinases to see how the compound works at the molecular level, to learn their binding modes, and then learn from that to see whether they can get a better compound. And based on this, all this screening, they found that the 7-aza-indole, they chose that as a scaffold. I'll show you on the next slide what that is. And then because this scaffold can be modified in many different places to give you different interactions with the kinase. And then they, based on this scaffold, they made many different analogs and then screened them against this mutant of RAF. So this is 7-aza-indole. This is indole 7-aza, means 7-nitrogen. So you can sort of see this compound bind to a kinase called PIM1. This compound bind to FGFR1, the receptor tyrosine kinase. You can see in this case, the 7-aza-indole make 100 bound to the backbone carbonyl and amide, just like N1 and 6. So now both 100 bonds are made. And this one here, this one's a protein. So the 100 bonds is not made because the protein is a protein. So there's no main chain nitrogen anymore. But anyway, so you can sort of see by putting in different substituents, you can achieve different interactions with different kinases. So that's sort of what they use at the starting point. And this led to this compound, PLX4720, and that binds in the region between the two lobes. You can see, again, 200 bonds made from the scaffold, just like adenine. And then they screen this compound against a panel of various different protein kinases to show selectivity. So against RAF, V600E, 13 nanomolar. Against wild-type RAF, 160, so about 10-fold selectivity between the oncogenic mutant and the wild-type RAF. And then against other kinases, much less active. So again, give you selectivity. So how does it work? It turns out actually the compound binds both the DFG in and the DFG out conformation. So But the DFG out is bound at the lower occupancy, so lower affinity. It prefers the DFG in conformation. And then DFG out is bound, but it's not as much occupied. So that's a figure I made myself by overlaying different structures. You can sort of see that DFG in is green, so phenylalanine here, that's DFG in. And DFG out is cyan, phenylalanine is over here. So you can see the compound actually is much smaller than what I showed you. Even though DFG is out, the compound doesn't go in there. The pocket is left open. It doesn't actually use it. So the compound stays where it is. And then this is the alpha C helix again. So that's what I showed right here. So this is DFG in, DFG out. DFG out, the phenylalanine is out of the way. So the big pocket opens up here, but the compound actually doesn't use it. So it's left open there. So that's a possibility as well. So there are many different ways of getting small molecule inhibitors. So then the question is, how does the compound select for the oncogenic mutant? Because as you saw, it's about tenfold more active against the oncogenic mutant. So this mutation is in the activation loop, which in the structure actually disordered. They can't even see it. But because it's activating, that means it promotes the DFG in conformation. So this enzyme may sample back and forth more. The mutation forces the DFG in conformation more than the DFG out. And then the compound actually has higher affinity for the DFG in conformation. That's sort of how, so the mutation makes the protein more DFG in. The compound is more selective for that. That's how you achieve that selectivity. So very interesting mechanism of how the compound works at the molecular level. How do you select? It's not like it binds to this mutation anyway. It's indirect. Okay, it changes the conformation. That's how it gives you a higher affinity for the activated mutant. So this ultimately led to this compound, PLX4072, which is this NIP compound that has been approved for treating melanoma. So you can sort of see a lot of work went into, a lot of screening, optimization, chemistry, biology went through, and then a lot of compounds get made as well. So this number here means the compound. And then in the end, in this case, it's a success story. You get a compound on the market. Okay? All right, so what's next? Oh, yeah. So another story, another kinase, more inhibitors. Oh, this actually not inhibitors in this case. AMPK, which you heard about on the top of fatty acid metabolism. So AMPK, as the name suggests, AMP-activated protein kinase. So 1-2 cells have high AMP levels when they have low ATP levels, because AMP and ATP are anti-correlated. When you have a high AMP, you have low ATP. When the cell has low ATP, you have low energy, and that's not good. So that's an SOS signal for the cell. It said, okay, I need more energy. So how do you get more energy? You turn off biosynthetic pathways because they consume energy. So biosynthesis, fatty acid, cholesterol biosynthesis, gluconeogenesis, protein synthesis. All of these require energy, so you turn them off. So that's what AMP does. AMPK does. It phosphorylates the enzyme in its pathways to turn them off. And then on the other side, you turn on processes that drive energy production. So fatty acid oxidation, as I mentioned, is glycolysis, glucose uptake, and then mitochondrial biogenesis, because that gives you more mitochondria to do beta oxidation or to do TCA cycle, give you energy. So the AMP essentially is a central regulator of all of these energy processes. As you can imagine then, what we want is an AMPK agonist. We want to turn on AMPK so that we start burning fat and then stop making fat. So that's contrast to other protein kinase. We're looking for inhibitors. Here we're looking for an agonist, which is much harder because inhibitors, you can use active site at the starting point. Agonists, you have no idea because agonists by definition have to be allosteric. So now how do you know that there's a pocket there? Secondly, if there's a pocket there, how do you know binding a compound there will give you activation? Oh, so this I showed you already. So AMPK is a hydrotrimer. So alpha subunit contains the kinase domain, 300 residues, plus other domains for protein-protein interactions. And then in humans, it's called AMPK. In yeast, it's called SNP1, but that's the kinase subunit. And then the beta subunit contains mostly protein-protein interaction, except in addition, there's a domain here called CBM, carbohydrate binding domain. It has a structure that it sort of binds carbohydrate, so that's why what it's called here. And then gamma subunit called SNP4 in yeast. It has 4 CBS motifs, cystathionine beta synthase, doesn't really matter. And you'll see this actually is a sensor for the nucleotide, ATP, AMP, whatever it is. This senses the nucleotide that controls the activity of the kinase domain. So in humans, there are 2 alpha isoforms, 2 beta isoforms, 3 gamma isoforms. So you're talking about 2 times 2 times 3, 12 possible combinations of AMPK in human cells. They all exist, but in different tissue combinations, different tissues. So agonist, so this paper back in 2006 was a breakthrough. So this is the first reported agonist of AMPK done by Abbott. So the A here means the Abbott compound name. That's how their compounds are named. So A769662, you can see they did a screen, again, high throughput screen, based on AMPK activity on the peptide substrate, and they look for a compound that can activate the kinase. So this is the lead they found, the hit they found from the screen. They did chemistry. They found this compound, which is 769662. You can see the compound actually is quite potent, EC50.8 micromolar against the enzyme from rat liver. Against the recombinant enzyme, alpha-1, beta-1, gamma-1 is about the same. And this enzyme, this compound is selected for the beta-1 subunit. Beta-2 is less sensitive to this compound. Okay. So that's the first one. And since then, many other AMPK agonists have been identified. For example, one from Merck, again, beta-1 selective, and then R734, R739. These are beta-1 and beta-2, so they're no longer selective for the beta subunits. Why beta subunits? Well, that's shown here. So here is the structure of AMPK bound to the compound, which is shown right here. 991, another compound. Okay. So here is the sensor, the gamma subunit. So it binds AMP and ATP at the interface between the motifs. You can bind 3 nucleotides, either AMP, ADP, or ATP. Then beta subunit sits right here that mediates the interaction between the gamma and the alpha. Alpha subunit right here, small lobe, large lobe. Yeah, this one's upside down, unfortunately. The kinase is upside down. Small lobe at the bottom, large lobe at the top. Okay? And then alpha C helix is right here. That's important for catalysis. And then interestingly, what they found is in the structure, the beta-1 CBM, that domain, carbohydrate binding domain, actually sits above the small lobe. And that creates a pocket. That's where the compound binds. So the compound's bound between the small lobe of the kinase and the CBM of beta subunit. And that's shown right here. That's where the compound is. And then the CBM, after the CBM, there's also a helix that sits next to the alpha C helix of the kinase. So that's how it additionally stimulates, because it helps to stabilize the alpha C helix to do catalysis. So the structure is quite useful in explaining how everything works. And also you see they have staurosporin bound in the active site. So that staurosporin is a non-specific protein kinase inhibitor. So it binds between the 2 lobes to stabilize the enzyme so that it can get a crystal structure. So that's shown better right here. So 991 in terms of polar interactions, many polar interactions right here, and then in terms of hydrophobic interactions, also isoleucines, phenylalanines binding to the compound. In addition, the reason is beta-1 selective because there's a serine residue from the CBM that needs to be phosphorylated. So once that's phosphorylated, it binds better. So that's phosphate recognized by this lysine side chain, ionic interaction. So how does this activation work? In addition to helping the active site, the other thing it may help also is avoid the dephosphorylation, the activation segment. So I told you that kinases need to be phosphorylated to become active. And there's, of course, phosphatases come in to remove that phosphate, to turn it off. So here they did assays. So PP2C is a protein phosphatase. So if you put in a phosphate, and then enzyme is dead because you dephosphorate the enzyme. And then AMP, ADP does not protect against the dephosphorylation. But the compound does protect somewhat, at least 50% or so. So that's what given this model here. So the active kinase had the activation segment with the phosphate on it, and then the protein phosphate come in, remove the phosphate, the enzyme is dead. And then when you have a compound present right here, the activator right here binds between the CBM and the kinase, that buried the activation segment. So now that phosphate is somehow protected against dephosphorylation. That's why you can get prolonged activity of the kinase, you get stimulation. So that explains that process. And then, so these are two other structures, R734, R1739. So this one's the right way up. Gamma subunit, beta subunit, alpha subunit, small lobe, large lobe, and then beta subunit CBM right here. And they are compounds bound same place between the CBM and the small lobe of the kinase domain. And then the compound actually, if you look at it, it binds quite similar binding mode. And in this case, they use a phosphomimetic mutation, changing the serine to a aspartic acid, right here, D108, D108. So they did not use a phosphate, they used a aspartic acid mutation to mimic the phosphorylation. Okay, what else is there? Okay, almost done. So last story, only a couple of slides. I'm sure you're tired of kinases now. CDKs, cycle-dependent kinases. So as the name suggests, it's involved in regulation of cell cycles. So different cyclins, couple of different protein kinases, control different stage of the cell cycle, as shown right here. Each stage of the cycle has a unique combination of a cyclin with a cyclin-dependent kinase, CDK. Okay. So CDK2 with cyclin is at the G1S interface. These are serine threonine kinases. And then CDKs alone are inactive, so it requires cyclin come in. The complex is the active species for this kinase. So this was done back in 1995, reported. So this is structure of cyclin A bound to CDK2, bound to ATP. So this is the original figure back then. That's the best quality we can get, I guess. So cyclin A is right here, all alpha helical. Protein kinase right here, small lobe, large lobe, alpha C helix. This is the activation segment. You can see ATP binds in the interface between the two lobes, and the cyclin bind near the alpha C helix, also the activation segment. And that's sort of how it activates the enzyme. So if you're doing an overlay, I think this is the last slide I have. If you're doing an overlay between the free and the bound CDK2. So here, red, yellow, and cyan are the cyclin A complex, which is not shown here. And green and gray is free CDK2. You can sort of clearly see in the complex with the cyclin A, you can see the alpha C helix moves much into the small lobe. So basically position the alpha C helix in the right place for doing catalysis. Same is true for the activation loop. In the free enzyme, it actually sits right here. It blocks substrate binding actually, and then in the active state, it gets out of the way, and then you can do catalysis. So there's another mechanism now to activate the protein kinase. You can help use the protein to position the alpha C helix, also the activation segment correctly. You get activity. So as I said, there are 500 protein kinases. There are probably at least 500 or more stories about how they are regulated at the molecular level. And many of these are drug discovery targets, and most of them use this binding site right here. Okay, so I think that's it. Yeah. So we have 1 hour in the afternoon to do PyMol again, right? So maybe I'll show you the DFGL thing later. So sort of give you a sense. Spend an hour to learn something. We can do morphing again, real morphing today. And then, so we can do that. And then for now, I guess we'll take a break. And then, I don't know what happens after this. You have tea time, is that right? Yeah. Okay, so tea time. Unless you have questions. But anyway, tea time. If I don't leave, you will not leave, is that right?